So Nikhil is a graduate from Glenwood High School.
You have to speak up. The microphone's not very
as
good.
number one, valedictorian, and then he's attending Case Western Reserve University in Cleveland for BSMD program, and it's one of the very competitive program in the nations. So when I talk with him about his interest, he's really interested to become a medical doctor and become an oncologist. I think his father had a huge influence on him. So he came to my lab doing work on this called link RNA. So link RNA is called a non-coding RNA. Usually RNA, messenger RNA make a protein, but this is non-coding RNA, about 1 kilobase long, but they don't encode protein, but they regulate many cellular process. And we found out this link RNA RMST can be stimulated when cells go encounter a metabolic stress, like hypoxia, glucose starvation, and mitochondrial defects. So in this project, Nikhil studied this RMST in glioblastoma cells. So Nikhil.
Hey everyone, this is my presentation titled The Role of Long Non-Coding RNA, RMST, in glioblastoma cells, and I'm Nikhil. So as Dr. Lee mentioned, LNC RNA stands for Long Non-Coding RNA, and it differs from typical RNA in that it does not code proteins, and instead it regulates gene expression and cell differentiation. So RMST is short for rhabdomyosarcoma 2 associated transcript, and it's a particular long non-coded RNA that I studied throughout my internship, and it regulates gene expression through interactions with proteins and other RNAs. So glioblastoma multiforme is a type of cancer of the brain and spinal cord. It is grade 4 tumors of the central nervous system, and it's highly aggressive and proliferative and associated with poor outcomes for patients. Studies suggest that RMST is involved in tumor cell proliferation. RMST regulates cell cycle control and apoptosis. RMST levels correlate with glioma aggressiveness, and RMST overexpression leads to tumorigenic properties. So what's the purpose? Why is it important to study this research? Number one, research on RMST overexpression in glioblastoma is crucial due to the limited amount of published research. This research is in its infancy. Through investigating this topic, we hope to understand the molecular mechanisms and pathways that govern glioblastoma progression and development. And we hope to see if RMST could be considered a potential biomarker for the early diagnosis of glioblastoma. Furthermore, if RMST plays a significant role in glioblastoma, we'd like to see it as a potential target for new therapies. And additionally, if there are different RMST levels for patients, we'd like to see personalized treatment approaches that allow for better treatment of patients. So I wanted to take a step back and just give you a background of what I learned in working in a wet lab environment throughout the last 9 weeks. So first, I was introduced to the aseptic technique, which refers to a set of practices and procedures centered around preventing contamination in the lab. This technique is essential for ensuring the integrity of lab experiments. And then within this technique, there's sterilization, which ensures that equipment and media are properly sanitized and we can work without contamination. And I also learned how to work in the sterile field, which took me a few weeks to learn. But working in the sterile field consists of working with sterile products, wearing protective equipment, and just following strict procedures to maximize lab results. I also learned how to culture cells, which is the process of growing cells in controlled conditions rather than than their natural conditions, which is in the human body. I learned how to revive and freeze cells. Reviving cells is the process of thawing cells to bring them back to active state, while freezing cells is the process of storing cells at very low temperatures for future use without compromising viability. I also learned how to both passage and seed cells. These processes involve storing cells for future use. Sorry, I misspoke. These processes involve moving a few cells into a new vessel with fresh medium to prevent overgrowth and maintaining healthy cell lines. And I learned how to prepare media for specific cells. Certain types of cells need their own nutrients and need to be in the optimal growth conditions. And then finally, I learned how to count cells. Counting cells determines the amount of cells within, the concentration of cells within a culture, and is important for standardizing experiments. And then in the realm of microscopy, I learned how to observe cells under a microscope. I learned to look for morphology, growth patterns, and responses to treatment. And I also learned how to stain cells. Staining is important for highlighting, staining cells with particular dyes is important for highlighting components or structures of interest. And the importance of these techniques is that I won't need a lengthy learning period when I pursue wet lab research in the future. I'll just be able to dive right in. So throughout my internship, I studied two types of glioblastoma cells, 2A1, which is on the far left, and 2C3, which is on the far right. And to determine the level of RMST expression within these cells, we did a qPCR, which is short for quantitative polymerase chain reaction. And it's a lab technique that allows us to measure the quantity of DNA in a sample. Fluorescent markers are used to detect DNA amplification as it happens in order to determine the amount of target DNA. So as you can see, there's a significantly greater amount of 2C3, which again is on the far right. There's a significantly greater amount of RMST expression in 2C3 than in 2A1, meaning that I can compare the 2C3 and 2A1 cells to determine what the role of RMST was in glioblastoma. The middle cells show 2A3, which is also a type of glioblastoma cells with RMST overexpression, but it was not a focus during my internship. So a little bit about the morphology, or like the cell shape of the cells. These images were taken with a microscope. So as you can see, the 2A1 cells appear more rounded, homogeneous, and densely packed, and there appears to be more of a random organization. In contrast, the 2C3 cells are more spaced out, they're less dense, and they have more of a elongated spindle-like shape. And additionally, you can see that there's more of an organization as the cellular processes seem to be extending towards each other. So during my internship, I performed an MPS assay to measure the rates of cell proliferation between the differences in cell proliferation between the two cell lines. So the MPS assay is a procedure in which measures the activity of mitochondrial enzymes and their ability to break down the MPS compound into a colored product. The amount of colored product can be measured by determining the absorbance of the solution. High absorbance values correspond to a greater number of viable cells, while lower absorbance values correspond to a fewer number of viable cells. While there was not a significant difference in the number of living cells on day 1, days 2 and 3 did have a significant difference in number of viable cells, meaning that the 2C3 cells had a greater proliferative activity, suggesting a possible result of RMST overexpression. And then I also determined the cell viability over a 3-day period. So for the general trend, you can see there's a gradual decline in both 2A1 and 2C3 cells. Initially, both were around a 98%, both had a cell viability percentage of around 98% on day 0. In comparing cells, 2A1, the 2A1 cells had greater viability each of the days, but this difference was minor. This gradual decline trend happens and is expected due to factors such as nutrient depletion, waste accumulation, and natural cell aging. And this minor difference could be due to genetic variation, again, RMST overexpression, that affects their resiliency to conditions. And then finally, measuring the cell viability is very important as it gives valuable information about the cell's health and functionality. During cancer research, scientists hope to screen for cells that are cancerous while preserving healthy cells. And then I also studied neurodifferentiation markers and their correlation with RMST. So as you can see in the bar graph, there's an upregulation of 4 different neurodifferentiation markers with RMST, and this suggests a link between the pathways that govern neurodifferentiation and RMST. Furthermore, there's additional evidence as the scatter plots also show a positive correlation between RMST levels and the neurodifferentiation markers, and these markers specify for astrocytes and neural stem cells. And understanding the relationship between RMST and the neurodifferentiation markers can shed light on the tumor microenvironment and help us understand how to disrupt these interactions. So this research, again, is in its infancy, and some ways that it could be advanced would be to study the mechanistic pathways undertaken by RMST. Additionally, clinical studies could be done to see the correlation between patients, between levels of RMST within patients and their outcomes. And then finally, in preclinical glioblastoma models, therapeutic strategies could be undertaken to determine the efficacy and safety. And thank you.
Questions?
When you were studying cell viability, what medium were you studying them in? Is it possible that the things could have different viability if they were still in a body, or?
I definitely imagine so. So the medium that we used was called MEM. It was tailored towards these glioblastoma cells. I would definitely imagine if they were in the human body, they would have different viability. But I also imagine that the ratio would be similar between the two sequences.
Yeah.
Just right here, just a quick question. Did you treat yourselves with anything or just?
No.
Okay.
Can you repeat the question?
Yeah, she asked, did I treat the cells with anything for viability or the MTS assay, and I did not.